Oh, why is this on side 3? Lol. Okay, mutations. Point and frame shift mutations. In case you don't know, a mutation is a change in the nucleotide base sequencing of DNA or any sort of like strand or template strand. And now a point mutation is a single alteration of a base pair, right? It's a single alteration of base pair, meaning that you're substituting a base pair in for one another. I did not mean signal, by the way, in the typing thing. I meant a single. That was a typo. Frameshift mutation is, however, is insertion or deletion. So, in quote unquote, it's changing the frame. Insertion is when you add a base pair, and then deletion is when you remove a base pair. Now, types of point mutations. Silent is when the amino acid does not change, right? It doesn't change. Um, and so it's when you substitute a base pair from one another, but the amino acid stays the same. Now, this is not really that detrimental because you're not really messing the transcript too much, and compared to the other ones, it's really not that detrimental. Missense is when the amino acid changes. So when you substitute a base pair, but the amino acid changes. This could be detrimental because it can mess up the whole entire uh, code, and it might make it untranscribable if you mess up the code, right? So this could be detrimental, and it's definitely more detrimental than silent. Nonsense is when it results in an early stop codon. I did not mean top codons, I meant stop codons. So when you insert a base pair, it results in an early stop codon. And there's three stop codons: UAA, UGA, UAG. There's a simple phrase to remember all three. You are annoying, you go away, you are gone, kind of representing that we want to terminate the sequence, right? No more. That's what a stop codon does. So it's an early stop codon, and this is very detrimental because you're essentially cutting the protein or the template strand. So this is extremely detrimental to the environment. Um, Amino acid. Okay, so yeah, if I had to rank in the most detrimental, it would be nonsense, missense, and silent. Here's just a picture of point mutations. As you can see in silent mutation, the amino acid stays the same, nonsense, stop codon, and then missense, different amino acids. Now a frame shift, like I said, I'm going to get a little more deeper, is a way of dividing the sequences. A frame, I mean, not a frame shift, a frame is basically the consecutivity of nucleotides, right? So, because nucleotides are in groups of three. They're codons, right? We're not going to be worrying about anticodons and tRNAs, that stuff. That'll be in later videos. But a frame is just basically the consecutivity of codons, and none of them overlap because they're in triplets, right? So none of them overlap. And frame shifts, like I said, I want to bring up a very important. They're insertion and deletion. But if you add a multiple of three codons to a template strand, then it won't be detrimental because remember, you're just if you have if you unless if you add a multiple of three in the template strand, then that's a codon, right? So nothing gets shifted. It just simply makes the codon longer. But if you remove it, it makes it shorter. So it doesn't have that much a detrimental effect as in multiples of one, two, or five, or whatever. So if it's in multiples of three, it doesn't have that much of detrimental. So it's very detrimental unless in multiples of three, frame shift mutations. And multiple threes, I mean codons. So if you insert three codons or you delete three codons, it's not as detrimental as a regular frame shift mutation. These are just a picture for frame shift mutations. As you can see, you're inserting a uracil between the other uracil and the guanine. You get UUG. I actually don't know what that is. If you do, comment, but I don't remember the genetic code. And that's going to do it for today's video. It was a very short video, but we just covered mutations. And if I kind of talk too fast, remember you can always slow down the speed to like 0.75 so you can really absorb. But I think you have a good grasp now. Thanks for watching. Also, make sure to subscribe and share this video to any of your friends who you think would be interested.